So Nina holds a master's degree in botany from Claremont Graduate University and a Bachelor of Science with a minor in sustainability studies from SUNY Oswego. Did I say that correctly?
SUNY
Okay,
Oswego.
SUNY Oswego. Okay, sorry, I'm a little, anyway. She currently works at the University in Jepson, Urbana, at the University of California, Berkeley, where she's the managing editor of the Jepson eFlora. If you have not used the Jepson eFlora, it's just a wonderful resource, and it's quite a project, I'm sure, to keep that updated. So Nina is one of the main people behind that. And she's also the coordinator for Jepson's public programs. If you've ever had a chance to go on a Jepson workshop, they are also really wonderful. They offer several a year. And Nina was just telling me before 7 o'clock that there is going to be a new sort of long form video series, kind of in that same vibe, that are coming out as well. And so in her free time, we always ask speakers to give a couple of, just fun anecdotal things. In her free time, Nina enjoys reading, hiking, biking, yoga, crafting, and spending time with her friends, family, and cats, or just one cat.
Yeah,
And
one cat and a roommate's cat.
and a roommate's cat. And then on a more personal note, I had the chance to meet Nina last year on a trip to Berkeley, and I gotta say she has an excellent taste in picnic spots. If you spent any time on Berkeley's campus, you might know about the Crawdad Little Creek thing going on there. This is the first time I've ever seen crawdads in California, and it was very interesting to have lunch there. And so anyway, that's about it. Nina, I will turn everything over to you. And yeah, we're so glad to have you as the speaker.
Great. Thank you, Mitchell. I can't take all of the credit for the picnic spot. My coworker Avery likes to picnic there, and often when I'm stuck at my desk, she encourages the rest of us to go outside. Okay, so yeah, thank you again, Mitchell, for that lovely introduction. Thank you to the California Botanical Society for having me tonight. I'm really excited. This is a really fun talk to be giving. It's a combination of my current work in my current position at Jepson, and also work from my master's project. So thank you all for being here, and let's get into it. I will start with a general overview of the Jepson eFlora. What is it? Why is it important? How is it maintained? And then move into different types of studies that inform the eFlora, specifically floristic studies. What are they? Again, how they inform the eFlora, and then examples from my own floristic study that I did during my master's project at the California Botanic Garden. And then wrap it all up, because everyone loves an action item. I'll wrap it up with how you can get involved with the eFlora and all of that. Starting with where I'm based, I am based out of the University in Jepson Herbaria. So the Jepson Herbarium is part of that larger University in Jepson Herbaria. We're two collections of pressed plants that are housed together with research labs, libraries, and archives. Altogether, the two herbaria hold 2.2 million specimens, all of it located at UC Berkeley, up here in Berkeley, California. The Jepson Herbarium was established in 1950 after Willis Jepson left an endowment to the university and his personal herbarium collection and his specimens. So both herbaria are housed in the same location, but we just split out the specimens in the cabinets. The UC Herbarium came before the Jepson Herbarium. It's part of the founding of the university. So we'd like to joke that we're not going anywhere. We're in the founding documents. You're stuck with us, which is a good thing. So the two herbaria have different focuses. The Jepson Herbarium itself focuses on the vascular plants of California specifically. The University Herbarium has a broader scope worldwide, and everything that was traditionally considered plants. So that includes fungi, seaweeds, lichens, and then vascular and non-vascular plants. Okay. So what does the Jepson Herbarium do? Well, of course, we manage the Jepson Herbarium and all of the specimens that are a part of it. We also manage the Jepson eFlora and other associated online resources. We also have public programs. Many of you may know about us because of the Jepson Workshop program. So Stacy Marcos and I co-coordinate that program together. As well as some of our other public programs. We also manage the Consortium of California Herbaria 1. So there is a CCH1 and a CCH2. I'm not going to get into those differences tonight. If you have questions about that, totally happy to answer them. But just for the sake of this talk, know that we manage CCH1 and all of that data in CCH1 gets fed into the eFlora, and that's how a lot of the information gets into the eFlora, especially regarding ranges and these types of things. Many of you are botanists and have used the eFlora, but I did invite some non-botany folks tonight. So I'm just going to really quickly run through what a typical couple of pages in the eFlora look like. So if you are trying to key something out, you are basically doing a choose your own adventure, right? So you would say, I have a flower in my hand. Does it have pink petals or does it have yellow petals? If it has yellow petals, I'm going to go with 1 prime, and then I'm going to work my way down, answering different questions about the characters of this plant until I find the thing that I think that it is. So this is what we call a dichotomous key. Once we've figured that out, we'll click on one of the plant names, and then that will bring us to a taxon page in the eFlora. So this will give us information about what that plant looks like, including like maybe leaf characters, such as how long it is, what color it is it hairy or not, flowers, how many stamens it has, what color it is again, stuff like this. And then moving more into ecology, elevation range that it's typically found in, the bioregions that it's found in, etc. We also link to resources outside the eFlora, including the California Native Plant Society's Rare Plant Inventory. And oftentimes, we have illustrations that depict what that plant looks like. And we also have photos down at the bottom of that taxon page. These link to the photos that are in Cal Photos. So we really, if you have really great photos and they're not in Cal Photos, please upload them to Cal Photos. Then you have the option to have them appearing in the eFlora on these taxon pages. If you have a lot of photos that you are like, wow, that's going to take me a long time to upload them to Cal Photos, you can email me. We have a bulk upload system, and I can work with you on how to make that happen. We also have 2 maps at the bottom of each taxon page. The one on the left depicts the bioregions that plant is known to occur in, and these are dictated by the author of that treatment. And then the map on the right shows all of the herbarium specimens that have been collected of that plant. Each of these dots represents a herbarium specimen that is in CCH1 that has been identified to this species. So yeah, it's really, really great information on these pages. How is the eFlora maintained? So, great question. This wonderful group of people, we are my coworkers. We all work together to keep the eFlora up to date. So Jason Alexander is our biodiversity informatics manager. He is the one who is doing all of that hard coding that I don't know how to do, right? So he yeah, he's the one making the eFlora pages look pretty and keeping them in ADA compliance and managing all of that data that gets put into the eFlora. So he has a hard job and he's doing a really, really great work. Bruce Baldwin is, was the Jepson curator throughout his career. He retired last year, or I guess 2 years ago now, but has stayed on as convening editor and is also working really, really hard despite being in retirement. Him and I just worked together on getting the revision out for this year. So Bruce is very busy and we're really grateful to still have him involved with the eFlora. I am, again, the managing editor of the eFlora, and I'm also a co-coordinator for the workshop program. Avery Ligon is our admin assistant and my little desk buddy. And then Stacy Marcos is the associate director of the herbarium and also, yeah, again, co-coordinates the workshops with me and has been super involved throughout the entire Jepson Flora project. So that's all of us together in-house working on the eFlora. Everything that we do is based off of Willis Jepson's work. So Willis Jepson was the first botany PhD student out of UC Berkeley. He then went on to teach at Berkeley his entire career and focused his research on the California flora and documenting it, publishing books on it, educating others on it. So when he was getting later on in his career, he realized, wow, I am not going to finish this project. So he left an endowment to the herbarium, encouraging us to continue on in his legacy of teaching folks about the California flora, publishing resources on it, etc. So fun fact, the Manual of the Flowering Plants of California was one of his books, and this year is the 100th anniversary of that. So we're celebrating that this year, and we're also celebrating the 75th anniversary of the Jepson Herbarium this year. So that's the basis. And then in 1993, the Jepson Flora Project published the Jepson Manual. In 2012, we published the Jepson Manual 2. And then pretty much immediately after that 2012 book was published, we also did the Jepson eFlora. So we were able to, yeah, move online because, you know, that's a lot faster to keep, easier to keep updated, faster to do revisions and then publishing a book. So that's what all of our current work is based off of, those books, as well as herbarium specimens in CCH, as I've mentioned. Herbarium specimens will come up throughout this talk, and it's important to know that almost all botanical research is based on herbarium specimens. So I really can't emphasize that enough. We are 5 people. We are also 200 people. We really could not do the work that we do and keep the eFlora up to date in the way that we do without the, you know, the work of so many people, including a lot of botanical researchers throughout the state and even throughout the world. So these folks are all doing their own botanical research, publishing new names, finding, you know, new populations of plants outside of the known distributions. And we work with all of them to get the treatments in the eFlora up to date. And we are so thankful for them and working with us and everything that they're doing to better our understanding of California botany. One way that those folks are involved with the eFlora is through our annual revisions. So revision 13 just got posted to the website last week. If you haven't looked at it yet, and if you didn't come to revision 13 review workshop this past weekend, I don't know what else you could have been doing on Saturday, but it was actually a really, really great workshop and a lot of fun, and got a lot of good information about why these name changes happen, you know, and the work that these researchers are doing to inform the flora and these changes. So just what is an annual revision? So they used to come out in December. We are now releasing them in February of each year. So this is the first year that we did that, just because it fits a little bit better with our schedule. Hopefully it fits well with all of yours. So these revisions are informed by the most recent scientific evidence that these authors are publishing in the scientific literature, and it is meant to supersede what was printed in the Jepson Manual 2. So if you are going out in the field and using the second Jepson Manual, it's important to then come back and check out the eFlora and see how maybe names have changed or anything like that. The Jepson Manual, the book, is still useful and very important and a good resource when out in the field and, you know, not having access to the internet or something like that, but always important to check because a lot of names have changed in the, you know, 13 revisions that have happened since the book came out. So some of the types of changes that happen, some of the work that informs the changes that happen in the eFlora, I'm just going to run through three different kinds of papers that can result in changes. So this one by Bruce Baldwin, Susan Fawcett, and Scott Armbruster came out last year. So listing 3 new species of Collinsia for California. So this type of paper is this type of change is listing plants that are completely new. So it's not like basically what might have happened is Bruce and Susan are out in the field and Scott are out in the field. They see some Collinsias and they go, oh, those are weird looking. Those have different characters than any of the Collinsias we know. And then they look at the morphology more closely, maybe do some phylogenetics and realize, oh, these are completely new species, never before seen. So, or never before described to science, right? So this is one type of change that can lead to a revision. Another one is has been described in this paper that Barbara Erder and I published last year. I'm super happy and to be working with Barbara on this work, and really happy she involved me in this project. But this is listing changes so these plants already are known to science but need a new name based on phylogenetic work that has shown that Potentilla E, the way that it is currently defined, is not monophyletic. So you have Potentilla, Ibesia, Horkelia, Horkeliella, and maybe you have the way that the phylogenetic tree is falling out. You might have Potentilla names mixed in with Horkelia names, mixed in with Ibesia. So what needs to happen? Well, this is debatable. I know Barbara's on this call. But what is happening is all of those names are getting lumped together under Potentilla. So once this revision happens, we will no longer have Horkelia, Horkeliella, and Ibesia in California. We'll just have Potentillas. So this is one example of a change that's coming to the eFlora soon. And then another way that we can have revisions is through these really awesome papers that are noteworthy collections. So oftentimes what these papers look like is a plant that's already known to science, but we just didn't know it occurred in California. So sometimes this can look like, oh, there's something that's growing in Nevada, and then someone finds it just over the line in California. In this case, the folks at Santa Barbara Botanic Garden, Wildlands Conservation Science, USGS, US Fish and Wildlife Service, etc., found some new plants on Santa Rosa Island. So that means they're going to get added to the eFlora. So exciting. So that's all work that's being done by botanists and scientists in the field, but there's also a huge role that citizen scientists are playing, especially on iNaturalist. And this is just one example of that. So in this series of comments on this observation, the original poster said it was an astragalus. Someone came in and said it was something else. And then a whole conversation ensued where someone was like, no, I think it's this. And then they're like, that doesn't quite fit. And then they realized it was actually something that's not in the eFlora yet. So the result of that conversation is Kier emailing, well, saying, okay, I'm going to email Jepson and get that updated. So he emailed. It's really cool to be like, I'm emailing Jepson, and then they're emailing me and Bruce, you know? So it's cool. So he emailed me, and now Bruce and I know to get this name into the eFlora. So everybody plays a role in the eFlora and can contribute to us keeping it up to date. So that's something that we're really we want your help, and we want you to communicate with us. If you're ever noticing something not in the eFlora and wondering why isn't it in there, just send us an email. We might not know about it. Again, this is a community project, and we really support your participation. So why is the eFlora important? So at the core of the eFlora, it is the most comprehensive guide for California plant ID that exists. And why is that important? Because we cannot protect plants. We can't conserve them if we don't know anything about them. And the eFlora is how we know about plants, how we know how to identify them, how we know where they occur, what they look like, all of these types of things. So the eFlora is a really like important, plays a really important role in the conservation and preservation of plants in California and in the surrounding states. It also, the data from the eFlora and from CCH, the Consortium of California Herbaria, feeds into many other nonprofit organizations that also play a large role in conservation in the state. So that includes the California Native Plant Society's Rare Plant Inventory, the Department of Fish and Wildlife's Natural Diversity Database, CNDDB, also California Invasive Plant Council, CalFlora, and CalScape. So our data and the CCH data is in all of these resources. So without all of these things, conservation couldn't happen in California. So we're really proud to be playing a role in that. And it's really, really important that we're thinking about conservation and how to identify these plants, how to conserve them right now. So I don't need to tell everyone on this call, I'm sure you already know, climate change is happening. It's happening right now. Massive wildfires this year alone in Southern California, and in, many previous years, we've had a lot of really, really bad fires, and large wildfires happen for a number of reasons, but it is definitely a factor playing a role in these wildfires is climate change. So climate change is changing fire regimes, it's raising temperatures, it's increasing drought conditions. And there's also a lot of other more direct human activities that are having impacts on plants, including deforestation, pollution, introduction of invasive species, and more. So there's a lot of things that are really hitting our biodiversity right now all at once. And it's important that we're paying attention and also paying attention to how we can protect these plants as best we can. It's really important also when we consider the fact that we don't know everything that there is to lose. This is a map from the Consortium of California Herbaria depicting where all of herbarium specimens have been collected in the state. So, and it's also depicting these things that we call botanical black holes. So what is a botanical black hole? It is literally, in this image, a black hole where no herbarium specimens have been collected, or little to no herbarium specimens. So why is this important? Why to pay attention to botanical black holes? It's because if we don't know, again, where these plants are occurring, what threats they're facing, other like ecology of them, where they like to grow, who they like to grow with, all of these types of things, we can't even think about trying to protect them because we don't know anything about them. So filling in these botanical black holes in our knowledge of where these plants occur and ultimately informs our understanding of plant diversity, species ranges, and potential threats to especially rare plant populations. And again, doubling down on the fact that we don't even know where, like, everything that we're losing. So as an example, so there's extinction that has been happening in North America. Approximately 65 vascular plant taxa have gone extinct since the beginning of European settlement. Most of these extinctions have happened in the Western United States and especially in California. And you can see everywhere there's a white dot on this map represents a plant species that has gone extinct. But when we think about white settlers coming to the United States, coming to the East Coast, and how active botanists have been in the West, especially in California, we can maybe start to think there are probably more plants that have gone extinct on the East Coast, and there are probably more plants in the middle America that have gone extinct, but without the high degree of botanical work that's happening here on the West Coast, we don't even know what we're losing. And that's really unfortunate. I mean, maybe we don't want to know how much we've lost. Maybe that's too depressing, but, So, yeah, and again, thinking about how much unknown diversity there is in the state, since 2012, 227 new native plants have been added to the eFlora. So since the last Jepson manual book, and in those 13 revisions of the eFlora, we have added 227 new native plants. So defining native plant here, or new native plant, this includes range extensions, taxa new to science, and those taxa that are newly recognized but not new to science, such as names that have been resurrected with previous or novel circumscriptions. So, and this doesn't even include, there are still names that Bruce and I are working on getting into the eFlora. They've been described, or it's been made aware to us that they need to be added, but we just haven't. We're working on it. So the number of newly described taxa, this is not 227, but is probably pretty high. We did the math on this, and this 227 new native plants since 2012. That's about 15 new taxa a year added to the eFlora, which is great. Also, just to throw these numbers out, there's been 36 new naturalized taxa added to the eFlora and 7 new waifs. And tracking naturalized taxa is also very important. So, and among those new taxa that have been added to the eFlora, we also have new rare taxa. So these are examples just from this year's revision. Botrychium pedunculosum, which is has a California rare plant ranking of 2B.1. Claytonia pearsonii, subspecies yorkii, is a 1B.1, and Nemacletus twizzlemanii, variety botany womani, is 1B.1. So again, coming back to that idea of what there is to lose, and rare plants are definitely like low-hanging fruit. These are the things that are going to go extinct very easily without us even realizing it, and especially if we don't get them into the eFlora, because people won't be looking for them, they won't be trying to protect them. So really, we're really happy to have these taxa in the eFlora this year. Another, this is outside of, this is a different type of revision that can happen in the eFlora, and just something I wanted to mention super briefly before moving into the next section of the talk. So range extensions these can happen as part of revisions, but they can also happen as errata changes, which are just really quick things that I can do like on my lunch break at work. An example of a really cool range extension that happened recently is this Orthocarpus cuspidatus subspecies cryptanthus that Keir Morse found on Mount Pinos in the Western Transverse Ranges. So this is a crazy disjunct. It's really incredible. He previously had just been known from the Sierras, Modoc Plateau area, and then the eastern side of the Sierra, and then he found it all the way over on Mount Pinos. He sent it to me, letting me know that he had found that, and then I emailed Marguerite Weatherwax, who's one of our Orobank ACE experts, and also talked to Mark Egger, and they both confirmed, yep, that's it. So now we have that updated in eFlora, and you can see that it is a range extension and a disjunct population for this plant. So now people know to maybe look, in here for this plant, and also look around a bit more in the Western Transverse Ranges for it. So again, a really important thing to get into the eFlora. And also, can you tell that Keir emails me a lot? Both of my examples have been Keir. But that's great. We want people to email us. Again, please let us know when you find stuff like this, and we want to get it into the eFlora. Okay, so those are all different kinds of studies that inform the eFlora. Also, and citizen science things that inform the eFlora. And I'm just going to narrow in now on one specific type of study that is really beneficial to the eFlora, and that is floristic studies. So what is a floristic study? Floristics are thorough documentation of the plant life in a particular region. So basically what that looks like is you pick an area of land, you document all of the plants that occur there. These are specimen-based studies typically. So that means going out in the landscape, collecting pieces of plant, pressing them, and then making herbarium specimens, right? So really important, really the basis for all other types of botanical research, because it is the major production of herbarium specimens that are used in all other kinds of botanical research, including DNA extraction, which leads to phylogenetic studies, morphology studies, everything else. So yeah, very, very important. So yeah, major products are the voucher specimens, and then also the annotated checklist, which is basically just a long list of all the plants that occur in that region, with certain helpful information such as typical habitat type, maybe disturbances, elevation range, these kinds of things. I often get the question of how do people choose a floristic study site? So there's a lot of factors that can play a role in that. And at the end of the day, it's a very personal choice, because it's, you're spending a lot of time there. But I just wanted to list some of the things that I took into consideration. And I know that some of my friends who have done floristic studies have taken into consideration. So botanical black holes are definitely one of those major factors. A lot of people, of course, want to feel like they're contributing to like an empty space in our knowledge. Like we want to get out and we want to explore this place that's never been explored before, right? Which is of course not. It has been explored before, but not in this way. So botanical black holes play a role in choosing a study site. Also, but also resurveys are really important to do. So if someone did a floristic study in a spot 50 years ago, and then you come back and do it now, it's important to you can see potentially changes that have happened in those 50 years, including changes that might have happened because of climate change or direct human activity. So resurveys are really important. Also distance. So depending on where you're located, how easy is it to get to the study site? Accessibility. Do you want to be hiking or backpacking 30 miles into the study site every time? Maybe not, especially if you're making a lot of plant collections. So those are important things to consider. Geology. I really like granite. I have friends who really like limestone, So what kind of geology are you into? Habitat types. I mentioned it before the talk began, but I'm from upstate New York. So when I first moved to Southern California, I was like, I miss fresh water. And the Sierras were a good place to find some water. So, and then these last two are a little silly, but there are often folks who, when I started my floristic study, I had been in California for 2 years. So to say that I knew like where to go to find cool plants, I mean, I knew some places, but Naomi Fraga was my master's advisor, and she definitely played a big role in helping me choose a good spot. I know my friend Marty Purdy, Dean Taylor recommended to him his study site, and he ended up finding some cool plants too. So and that was Coyote Ridge area near Bishop. And also, this one is silly, likelihood of finding cool plants, right? Like you want to know you're going to go out and see some cool stuff and have some fun. So, and for example, I got to see Carlquistia murray, which is a really great plant. It's a 1B.3, so pretty rare, and is a monotypic, Carlquistia is a monotypic genus right now. Bruce Baldwin mentioned that might not always be the case, but for now it is, and it was fun to get to see, and I made a seed collection of it for the California Botanic Garden. So anyway, things to consider if you are at all interested in doing a floristic study. All of that taken into account, I chose to do my study in the Southern Sierras. In addition to all of that, Sierra Nevada is a hotspot for endemism and plant diversity in California. Even though it only makes up a fifth of the total land area of the state, it contains over half of the plants present here, including really cool endemics and like rare plants, including this giant sequoia you can see here with my dad. So let's narrow in. Here it is, that white dot. Let's narrow in. So here is the study site outlined in black, completely located in Tulare County, but close to Inyo County and Kern County. Closest town was Kernville, also the closest cell phone service. And then closest cities were Bakersfield and Ridgecrest. Zooming in further. So the entire study site is located on the Sequoia National Forest. Half of it was on the Domeland Wilderness. You can see that here pictured in this darker green. It's made up of two watersheds, the Salmon and Manter Creek watershed, pretty much split exactly down the middle of 25 square miles of the Salmon Creek watershed and 25 square miles of the Manter Creek watershed, altogether being 50 square miles. The highest elevation point was Soretta Peak at almost 10,000 feet, and the lowest, the Kern River and Salmon Creek confluence at 3,400 feet. So yeah, North Fork Kern River to the west, and then the South Fork Kern River peeking in over here to the east. If you're looking for a place to go camping, some of the prominent spots, Horse Meadow has a campground. Big Meadow is lovely, also has a lot of cattle grazing, but still a really pretty meadow. Soretta Peak is a great hike. Manter Meadow is an awesome spot too. So definitely worth checking out. And I'm in love with the study site, but I want you to be in love with the study site too. So I'm just going to show you some pretty pictures for a second. Here is the view from Big Meadow from the highest point in the study site, Soretta Peak, up where the foxtail pines grow. Horse Meadow, very lush, very green. Here's the Salmon Creek flowing through it. Again, there's a good campground near here. Manter Meadow is in the Domeland Wilderness, a good place to backpack into. This picture was taken in 2019, just a really great rain year. And this was the greenest I saw this meadow. It's just super lush, really wonderful. Salmon Creek Falls. The Salmon Creek Falls are the highest waterfall, I believe, south of Yosemite. Definitely the highest waterfall in the southern Sierra Nevada. So really dramatic, really beautiful. The Salmon Creek itself drops over a mile in elevation across its whole range, and it's only like 12 or 13 miles long. So that's pretty incredible. A lot of that elevation is lost here. And you can see this from the highway that comes north of Kernville. It's pretty prominent. North Fork Kern River, really, a lot of river rats down here. It gets hot in the Kern Canyon, so it's a good place to hang out, drink a beer. Domeland Wilderness is so named for the granite outcrops that are found throughout the wilderness area. So Church Dome is one of those prominent ones, good for popular with climbers. Rock House Peak is another gorgeous one. Also, Rock House Basin is similarly named nearby, and the PCT runs through there. And then White Dome is just another granitic dome that, is an incredible, really beautiful, dramatic thing in the landscape. So a lot of fun. Hopefully you are convinced that it's a gorgeous, beautiful, wonderful area. Other folks are also. So the people over at the California Wilderness Coalition, and back in 2017, wrote up this paper to try to get the Salmon Creek labeled eligible to be wild and to be labeled a wild and scenic river. So this would give additional protections to the creek. And then this did pass. It has been labeled eligible, but now it has to go through Congress. And last thing I knew, I had to go through Congress. And as we all know, Congress is very busy right now. So hopefully that happens soon. But in the meantime, I'm grateful that extra protections are being considered for this area of California that I really love. And especially because this area does face some risks and threats. So this next 3 photos is aerial over the study site. You can see Big Meadow here. You can also see kind of Manter Meadow here. And this is the South Fork of the Kern River, which definitely pay attention to that one. So 2019, again, was a super, super great rain year, super bloom, all of that. Naomi and I tried to backpack in from over here, and we just could not cross the South Fork of the Kern River. We got stuck, and our backpacking trip was a lot shorter. But anyway, so all of these next three photos are around the same date, 2019 through 2021. So here's 2019, 2020, 2021. So drought is a thing that we know about here in California. It happens, but it does have inevitable impacts on plant life. There's going to be less annuals that come up in these years. And if we have continued drought, it's going to have impacts even on perennials and everything, all of the plants. So the series of photos is very dramatic. So just showing you that one more time. As a result of drought, there is a higher risk of wildfires, and this area does have a history of wildfires. So the McNally fire of 2002 was the largest wildfire of the 2002 fire season, did get into the study site a little bit in the Northwest. And then the Manter fire burned most of the Domeline Wilderness really, but definitely almost all of the Domeline Wilderness that occurs in the study site. And 20 years out, it was still very noticeable on the landscape, the result of these fires. So if you go just south of the study site in the Domeland Wilderness, or I guess this is just outside the Domeland Wilderness, but this is all Jeffrey pines. And once you are actually within the burn scar, the recruitment of Jeffrey pines that I could see was very, very low. I think folks need to do more long-term studies to see over time how these Jeffrey pines are coming back after the fire, but it does seem like there's a little bit of a type conversion happening. And the Domeland Wilderness side of the study site is very desert influence, so it doesn't seem outside of the realm of imagination that the drought and the fires and everything are having like a large impact on the vegetation types that are in this area. Some additional threats facing the flora of the Manter and Salmon Creek watersheds are more direct human caused. So there's cattle grazing, both fenced in and free range. There's also logging that was seen during the study. So I think this was probably canopy thinning, but even still, like that is beneficial, but walking around in the understory, the impact to the understory is very substantial in these situations. Just completely, tracks and everything ripped up, and there's definitely impacts from logging, of course, and so interesting to see that firsthand. And then there's also recreation impacts in the study site, a lot of hiking. There's some biking, including motorized biking on some trails, camping, of course, hunting, fishing. I ran into Danny Wagner, who lives in Kernville, and he likes to bring his goats up and do have them carry in his gear to go fishing in the Manter Creek area. So fun to see the goats on the trail. So just generally methods related to my specific floristic study. I began by drafting a checklist from historical specimens. Most of the fieldwork that occurred during this study was between April and September of 2020 and 2021. So most of the collecting that did occur was opportunistic, but there were some targeted searches specifically for rare plants or things I was hoping to relocate or things I suspected might be in the area. Detailed notes were recorded with each specimen. Most identifications were made using the Jepson eFlora, the Flora of North America. And then all of those specimens were deposited at the California Botanic Garden, Cal Academy, and the New York Botanic Garden. And here are some of those specimens. So super exciting. All of them have been deposited. They've all been accessioned. Well, I'm not sure what's happening with the New York Botanic Garden ones, but they're accessioned into their collections. And it's exciting to see all that data getting into the eFlora, into CCH. And yeah, really, really happy. So anyway, there's some examples of some specimens. And here is the culmination of all of that work also. So here you can see my specimens are where I made collections in white, and then where historical collections were made in black. Most of those historical collections were done by Ernest Twiselman, Gordon True, John Thomas Howell, mainly those folks. So I tried to fill in some of the gaps that they had not gone to, including off-trail, Domeland Wilderness in general. If you're looking for a place to do a floristic study, the Domeland Wilderness is a huge botanical black hole. So I really tried to get into some of these off-trail areas that were harder to access because they're rugged and covered in montane chaparral post fire. So all in all, I added 1,411 collections to the area, which more than doubled the number of collections in the study site. So bringing together those two pieces of the talk, how do floristic studies inform the eFlora? So one way, first let me introduce you to Eriogonum breedlevii variety Shavakii, which is the needles buckwheat. It is a rare plant with a ranking of 4.3. Previous to my study, it had been collected in the Needles area. Jim Shivak had done some collections of it up in his old study site. There were also some in this area over here, but all of these, except for maybe one, are my collections. So it had been previously known from the Southern Sierra Nevada High, which is the bioregion that the study site occurs in, but it had been a little bit disjunct in that these populations hadn't been connected. So especially with something that's a 4.3, which are taxa that we're looking to get more information about, because we don't know if they actually should be ranked higher in the, or I guess lower in the rare plant ranking system. So filling in our understanding of where this plant occurs, the types of habitats it likes to occur in is super important. And then Travis Columbus was with me during some of that collecting. So he was able to get tissue samples and all of that, which is, he's doing phylogenetic work on a lot of Eriogonums right now. So this was important work. Another way that floristic studies can inform the eFlora is through range extensions. So Gentiana newberrii variety Tyogana, you can see it has collections all throughout the Sierra Nevada. But you'll see this little one right down here. So that's mine. And this was a new southernmost occurrence for this plant. Why is that important? Plants populations at the edge of a species range are the ones that are most likely to be impacted by a changing climate, right? So especially the southernmost ones. As you have a warming climate, you have plants moving north, following the climate that they like to live in, or are adapted to living in. So the ones at the southernmost are going end of the range are the ones that are most likely to be extirpated first, or they also have potentially genetics that are better suited for that hotter temperatures. So looking at like how they're able to handle it and seeing how those populations are doing is super important to our understanding of how these plants are going to adapt to a changing climate. So yes, finding that new southernmost occurrence was awesome. I think this was about an extension by like 30 or 40 miles or something like this. So pretty exciting. This was right in Big Meadow with all the cattle grazing and everything too. So pretty cool to find it. On a similar note, Pinus balfouriana subspecies austrina also has its southernmost occurrence in the study site at Soretta Peak. This is one of my favorite plants in the study site. Really just awesome to see. These plants are really, really beautiful, really, really fun. But yeah, trees are even more likely to be impacted by a changing climate. Something we already know, as there's been very good documentation of trees dying off in the Sierras as a result of both climate change and pests and all these other things. So checking on this population was something that was really important to me. And I am happy to say that it was doing pretty well, actually. Besides some yellowing of needles, probably due to drought stress, this population was doing well. There was active recruitment of young individuals, and you can see that it is the southernmost occurrence here. And actually, it doesn't have that many. There's a lot of, or a good amount of iNaturalist observations of it, because you can get up to the population fairly easily. There's a nice trail, but there's not a whole lot of herbarium specimens of this plant, just mine and then one other one. So at least in CCH1. So yeah, happy to have documented this plant and gotten more information on it. Another way that floristic studies inform the eFlora is by documenting the spread of invasive species. So these three taxa, Myosotis discolor, Cerastium fontanum, subspecies vulgare, and Stellaria media, are all invasive plants in California that do not currently have Southern Sierra Nevada high listed in their bioregion string. So the latter two did already have specimens in the Southern Sierra Nevada high, but they don't have that bioregion listed. So that's something that Bruce and I need to work on updating. But Myosotis discolor didn't even have specimens previously in that bioregion. So, and it's actually kind of a crazy find, really. So there were a lot of there's a lot of documentation of it in Northern California, and also some documentation of it in the peninsular ranges, but there was nothing in the middle connecting that. So you can see my collection right here, smack in the middle. And now we will know that to add that bioregion to this plant's bioregion string, and perhaps look around more in the Sierras and nearby to fill in our understanding of where this invasive plant is occurring. No heart around this one, just a circle. Floristic studies can also bring attention to needed name changes. So this is one example of that. Abies magnifica variety Critchfieldii is endemic to the southern Sierra Nevadas. But when I found it, I thought it was Abies magnifica variety shastensis, because in the eFlora, that's what it identified as. So I uploaded it to iNat, said it was variety shastensis, and then it was quickly corrected by Michael Kaufman and Andrew Tree. And, as a master's student, I was like, what are you all talking about? That doesn't even... Variety Critchfieldii is not in the eFlora. What is this plant? And they very kindly sent me in the direction of this paper by Ronald Lanner, which described this new variety of Abies magnifica from the Southern Sierras. So then when I started working at Jepson, I mentioned this to Bruce and said, hey, maybe we need to get this into the eFlora. And he said, yeah, let's do it. So this was really like a full circle thing for me. I was really excited to have learned about this during my study, and then to be able to get it into the eFlora with Bruce and work directly with him on that. So it felt pretty cool. So just to show the differences here, Abies magnifica variety shastensis occurs in the Klamath Ranges and High Cascade Ranges. Abies magnifica variety Critchfieldii is the southern High Sierra Nevada. Both of them have these scales that are protruding, right? I think I'm using the wrong word, but you see what I'm talking about here. And then in the middle, the typical variety, Abies magnifica variety magnifica, does not have those scales. Or bracts. They don't have the bracts coming off of the scales. So it really is pretty well separated between the two. And yeah, now you can use the eFlora to figure out which variety you have. The floristic studies also bring attention to hybrids. So I found one putative hybrid in my study site. So Diplochus bicolor and Diplochus mephiticus have a known zone of contact south of Mount Whitney. And there have been hybrids documented in this area before. Naomi Fraga and I were out in the field. We found this plant, and she said it was one thing. I brought it back to the herbarium, tried to key it out, got really frustrated dehydrated. And then brought it to her and I said, Naomi, it is not what you said it is. I can't figure this out. She helped me with it and we figured out that it is a hybrid. So this is we do have hybrids in the eFlora, especially in oaks. So as we're, finding these things in the fields and we're finding more and more hybrids, and we can say that, okay, this is definitely occurring, this is definitely out there, we can add these things to the eFlora. So this isn't in the eFlora yet, but I should talk to Bruce about it. And actually we don't have to because he's here. He's hearing me talk. So Bruce, we should talk about this. Annotated specimens is another way that that is a super important contribution that floristic studies have to the eFlora and to botany in general. So throughout my study, I annotated approximately 250 historical specimens. I prioritized those taxa that were not relocated during the study. And how does this help? Well, it confirms. It's a way of confirming the identity of these specimens by having someone come in again, look at them, say, okay, yes, it is that, or, oh, no, it's not, and then re-identify it. That data gets into CCH, into the eFlora, and ultimately we end up with a cleaner data set. So doing annotations is really important. You don't need to be just doing a floristic study to do annotations. Please come to the Jepson Herbarium or CCH and feel free to, correct, confirm, etc. Okay, wrapping it up. I know it's getting late. So how are we capturing all of this new data in the eFlora? Well, I already said that, but revisions is one of the major ways. Also errata. So a lot of those things that I just talked about are little changes, that I can do as errata. So these are just things I take out the eFlora, I update the bioregion string. Maybe sometimes there's a typo or something like this. We have over 6,000 native taxa in the eFlora and over 8,000 when you include the naturalized taxa. There are going to be typos. So I can fix those pretty easily. And if you ever see something like that, definitely feel free to let us know and we can get that fixed. Again, community feedback is very important. I'm not going to go into this right now. I don't have time to do it, but one way that we are keeping track of range extensions and, plants that are outside, specimens that are outside of the known range of a plant is through yellow flags and purple flags. And you can see that depicted here. Again, not going to go into it right now. I don't have time, but just to say, for those who know about this, this is a way that we're tracking plants outside of their known range. And I'm happy to answer questions about this. This is a big project that we're working on, so definitely let me know if you have questions. Action items. I love to end presentations on an action item because it always makes me feel, woohoo, okay, something to do now. Make vouchers. I really cannot, do it with a permit in a way that is sustainable and not drastically impacting plant populations, but you have to make plant vouchers. iNaturalist is incredible. It's a really wonderful resource. It's not enough. We need to have that physical proof of a plant specimen so that we can do DNA extractions, so that we can get closer in on that morphology. We need plant specimens to really confirm that a plant occurred in a specific place at a specific time. So make vouchers, get that data into CCH. That is ultimately what's going to inform the eFlora. Anytime we have to do a revision and we say, hey, someone found something weird on iNaturalist, the author always comes back and says, okay, I need to see a specimen. So definitely vouchers, vouchers, vouchers. Again, I can't say it enough. Tell us about range extensions. Tell us about new taxa. If you're wondering why a plant's not in the eFlora yet, just email us and ask why. We might not know about it, or we might know about it and we just are working on it. So, yeah, just reach out and ask. We're friendly people, and we want to. I like talking to you. I like getting emails. You can use this QR code. There's links on every page of the eFlora that you can use to submit feedback when you find an error or something. So you can either email me directly or you can use that link. And that's it. I know I went a little bit over time. Thank you so much for listening and for hanging out with me tonight. And yeah, I'll take questions now. Hey.
Fabulous talk, Nina. Thank you. Wow. Wow.
Hey.
I I I learned a ton. Um,
Oh, good.
and yeah, your study site really isn't so far from home for me too. So yeah, you mentioned
Yeah.
that. Like that's pretty cool. So it is you are a little over time, but
Yeah. Sorry.
people who it's okay, that's okay. But people anyone who would like to ask questions, absolutely can ask. As long as Zoom doesn't kick us off. My computer died during your talk, so I am no longer the host.
Oh,
And I also some people did drop some questions in the chat, which I no longer can see. Maybe you want to talk about, go into those if there's time. Otherwise, I'll just
yeah.
open it up. If anyone wants to ask a question live, please do so.
Yeah. Well, maybe I'll have Peter ask his question and then I'll do chat questions if that's okay.
Thanks for the inspirational talk, Nina.
Yeah.
So I was wondering, did you um, kind of notice any effect of grazing on the floristics in your study site, I mean, just an overall reduction in diversity, or were there particular kinds of plants that were eliminated?
Yeah, great question. So Big Meadow has a pretty long history of grazing at this point. There's a really good quote. Joe Fontaine has a book called The Kern Plateau, where he has this really great quote where it's like some folks who were coming up from Bakersfield heading to Mount Whitney went through Big Meadow and they were like, wow, it is bad here. And that was in like early 1900s, 1800s. As far as like specific plants that I've noticed be impacted by it, it's a lot of like tops, like they're eating the tops of it. It's like the carex and the grasses. I did find Fritillaria pinetorum at the edge of Big Meadow my first year of my study, which is a rare plant. And then the second year it wasn't there. And it was, same spot, but there's a lot of cattle activity right there. So they could have been munching on it. I don't know, but this is a good question because I have a side project where Laura Cunningham and I are Laura Cunningham is the director of Western Watershed Alliance. And she's been looking a lot at the cattle grazing happening in the Sierras all over the mountain range. And her and I are going out to the study site, and we did a early season look at how the meadows look, and we're hoping to go to back in the fall this year to see late season how the plants are looking after a full season of grazing. And then we're planning on writing a letter to the Forest Service and right with the management recommendations and all of that. So I really will have a better answer to your question once that letter is written. Um, and yeah, but yeah, in the meantime, there's stream, when there's cattle grazing, like the stream banks aren't as steep. They're often the cattle like wear them down over time, so it affects stream health. Um, and you can see that Manter Meadow has no grazing, and the super steep stream banks, like really clear water. There's a variety of golden trout that occurs in that stream, really really beautiful. And then the Salmon Creek is just really impacted by grazing. There's no golden trout there. It's just like trampled. All the springs are trampled. Yeah, and there's definitely very visible impacts.
Thanks.
Yeah. Yeah. Okay. Um, let's see here. Benjamin says, visiting Death Valley years ago, there was a truncated version of the Jepson tailored to Death Valley. Is there hope on the horizon that someday folks will be able to filter the Jepson to, say, the Klamath Range and have keys for specifically that region and the flora within? Trying to cheat my way through agristology and willows. Yeah, I totally get it. So, there is something that exists like that right now called Keybase. Keybase is out of date though. We gave Keybase our data so that they could create Keybase. But then since then, Keybase can no longer be updated and it's not owned by us. So you can still use Keybase, but just know that recent revisions are not in Keybase including like things like Phrymaceae and stuff like this. So I think for willows and grasses, it probably is mostly okay to use, but for things like monkey flowers, like I wouldn't recommend it. We are definitely hoping to do something similar on our end. We have uh, right now, Jepson or Bruce is still working with us as the convening editor, but hopefully within the next few years we'll be able to let Bruce move on into his retirement, and we'll hire a new convening editor and Jepson curator who will definitely take the lead on something like this. We know people want offline access to the eFlora, and we know people want to be able to filter the keys down. So great question though. We really good. Andy Simpson says, you mentioned range extensions being possible as a result of revisions or errata. What about range extensions as a result of climate change? That is a great question. I cannot think of any examples of that right now, but definitely as things move around, I can see us having to, I mean, it might be hard to differentiate if something is moving, if it's always been there, or if that thing is moving because of climate change. So yeah, I mean, one way that we track like things being outside of their currently known distribution is with those yellow flags, with purple flags. Purple flags represent things that are in horticulture. We have things like Lupinus arboreus and Pinus radiata, and now Malva surgentiflora. These things are native to California, but are very weedy and are naturalizing outside of their known range. And one thing that we're considering is changing the color of the flags that are outside of that known that range that was where they originally occurred, right? So like Malva surgentiflora has one of those like subspecies is only known from like one of the Channel Islands, and now it's naturalizing on the mainland. So changing the color of those ones on the mainland so that we can know that those are not native, right? So that might be an approach to range extensions that are a result of climate change, like noting that these things are moving not because of outside factors, but definitely open to ideas with that question too. But it's a good question. Thank you. Okay. Andy Simpson also asks about the bristlecone pine. It doesn't live quite as long as Pinus longeva, but it still lives a long time. I would predict that it would have evolved more resistance to climate change purely because of its lifespan, in which individual trees must survive climate changes, not to mention populations. Do you see other species of tree being more responsive, vulnerable to climate change than Pinus balfouriana? Yeah, good question. I mean, bringing it back to, well, so Pinus monophylla, 100%. So one of the other graduate students out of the California Botanic Garden did a floristic study just south of my study in the Kiava Wilderness. And literally during her study, she had crazy drought years, and she watched the Pinus monophylla individuals die over the course of like 2 or 3 years. So this is something, and then I've talked to other folks who live or recreate near near right around the South Fork of the Kern River, Kennedy Meadows. And the person that I spoke to at the time, he was like, yeah, I've watched Pinus monophylla die out along the South Fork of the Kern River here. So they are definitely being like very heavy hit by climate change. The Jeffrey pines, just to note that they're not recruiting in the study site as much, is something to pay attention to. I can't say for sure that they're being really heavily impacted by climate change, but they're one to pay attention to, at least in that region.
Pinus monophylla is a pinion, which means that it's more related to Pinus edulis and the like, which also had a massive range contraction as a result of a super drought.
Yeah. Cool. Interesting. Well, not cool, but interesting. Yeah.
I would expect those species to be more mobile though, since pine crows do a lot of their dispersal.
Mm-hmm. Yeah, good point. Yeah, cool. Yeah, good questions. Those are great. Thank you. Victoria says, I have a question regarding your study area. How many acres approximately, and are you planning on possibly creating a field guide for the area? Approximately 50 square miles. I am sorry that I cannot do that conversion to acres in my head. But creating a field guide, that sounds awesome. I'm currently in the process of getting it published in ELISO. It has been submitted to ELISO, and I'm waiting to hear back from reviewers, hoping to get that published by the end of the year. So, so that's the publication, the manuscript. A field guide, that would be really fun. I think would require doing a few more trips to the study site to get more pictures. But I would love to do something like that. I definitely dreamed about it during my project, but got swept away in getting the publication itself done. So, yeah, if people want it, I can work on it. Marianne says, where can I find out what information is required for a voucher? Ooh, good question. So there's a lot of resources online about that. I think we might have something like that on the Jepson website. And that would be something that you could find by searching like what goes on a herbarium specimen label, right? And these are usually things like the identity of the plant, who collected it, where it was collected, which includes like latitude, longitude, general like 5 air miles north of Kernville, like that kind of information, associated species, disturbances, these types of things. So, yeah, but all of that is definitely online. And if you email me, if you're having trouble finding it, email me and I can give you a resource on it. Oh yeah, Tony, thank you. Yeah, CNPS, CNDB, those are great resources. Yeah, bryophytes. I know. There's a few other students at CalVG who have done bryofloras also. I felt like I was like, oh man, learning the vascular plants is hard enough. But it would be cool to know the bryophytes at the study site too. Something to do when I have more free time, maybe. Yay, okay. I think I did everything. But yeah, Mhm.
Yeah, there have been right in Madroño, there's been some fascinating bryophyte work that's come out recently. I can't remember the author's name right now, but anyway, Nina, thank you. That I think we might call it.
Okay.
This
Yeah.
this talk will be available on our YouTube page. Thank you again so much. And yeah, if anyone wants to continue the conversation, please email Nina. And yeah, that was fantastic.
Yay. Yeah, thank you everyone. Yeah, thanks for sticking around so late. Yay. Definitely feel free to email me. Yeah.
Thanks, Nina. Well, have a great night. Have a great night, everyone.
Okay. Bye.
Bye.